Hi everyone, welcome to your lecture on the cell cycle and mitosis. Mitosis is the process of cell division. It's how cells make more of themselves. So we talked about in class, the question here is, how do little elephants grow up to be big elephants? We asked the question, why, when you get larger or an elephant, do they get larger cells or do they get more cells? Talked about the surface area to volume ratio and how cells need to get nutrients to the center of the cell, and they can do that more efficiently if they are small. So they don't get larger cells, they get more cells. So a large elephant has a lot more cells than a little elephant. Why do animals shed their skin? The process of asexual reproduction begins after the sperm fertilizes an egg. Okay, asexual reproduction means not sexual or non-sexual, and that is the process of mitosis, where an individual cell reproduces and makes like two identical copies of itself. So that happens directly after the sperm fertilizes the egg. That actual fertilization process of the sperm and the egg uniting, that is sexual reproduction. But after that, when that cell, that single cell that we all begin from, divides, that is where mitosis begins, that first division. And we all start as one cell. And then through mitosis, we become everything that we are right now. So it's pretty amazing. So 3 reasons that cells reproduce by asexual reproduction are growth, repair, and replacement. So you get more cells through mitosis when you're growing. My 3-year-old daughter and my 8-month-old son, they are a lot smaller right now than they will be when they are full grown, and they continue to grow by reproducing more cells through mitosis. Repair. If you have a cut or something like that and you need new cells to heal that area, then repair is going to happen also through mitosis. And replacement, you lose cells all the time through the natural aging process or dying. Sometimes when you get sick, the cells will die or be destroyed by viruses or bacteria. So all sorts of reasons that cells die and they need to be replaced. You have over 1.2 trillion cells in your body, and they are constantly being replaced by new cells. So growth, repair, and replacement are the 3 big reasons for mitosis. Mitosis also is the cause of cancer when it goes wrong. A mutation during mitosis will form cancer cells or cancer cells. So skin cancer is the abnormal growth of skin cells, most often develops on skin that has been exposed to the sun. And cells that reproduce by asexual reproduction reproduce constantly. Majority of the time when cancer forms, there has been some sort of injury to that area. So skin cancer, if you have a lot of sun exposure, then you're damaging those tissues over and over again, and then cancer forms in that area. And we watched a video about that by Ava Vertes, and what the newer items are as far as discovery of the causes of cancer. Cell division is a fascinating biological process, and it's the building block for complex life. The amount of time it takes for cell division to completely take place ranges widely, depending on the species involved. Some bacteria, for example, can complete cell division in 20 minutes, while mammals such as humans may take 12 to 24 hours. This is a reflection of the complexity of cells, but also the purpose of the cell division. Bacteria need to divide rapidly to reproduce and grow, while adult humans are not in a growth stage and therefore do not need to produce as many new cells. I have a root tip right here, okay? So this is a model of a root tip and a bisection. And if you look here at the very end, the very tip, this is the area where the root is growing. So this part at the bottom here at the tip, that's where most of the mitosis is going to be occurring, where new cells are going to be forming as this root gets longer. Okay? And up here, they're pretty well formed, so mitosis doesn't happen as rapidly. It would be more for repair and replacement instead of growth. All of the growth is happening at the tip, so this is where all of the mitosis is going to be happening. There's different rates of mitosis depending on the stages that of growth they're in. So this circle of the cell cycle shows on the outside ring G1, S, and G2, which are the phases of interphase. G1 is when there is normal growth and development, so it's when the cell is just doing what the cell is supposed to do, whatever it's been specialized for. S is the synthesis phase, or when the DNA is duplicated. At that point in interphase, once the DNA has been duplicated, the cells are going to go through mitosis. There's no turning back. That is the no turning back phase. And then G2 is preparing for cell division because the cells have already produced a copy of the DNA. Then that they're going to need to also copy the organelles and grow and prepare to completely split and make two cells. And then the nuclear division point where it says M, that is the whole thing of mitosis. Remember, prophase, metaphase, anaphase, telophase. Mitosis is nuclear division, which means a division of the nucleus. Nuclear is nuclear division. And what is found in the nucleus? DNA. DNA in the form of chromosomes it's supercoiled genetic information. So they look like this is a electron micrograph picture of DNA. They look very fuzzy. I mean, that's a ton of genetic information that's just super coiled and very, very condensed and into a chromosome, so it's easier to split. Chromosomes contain genetic information, or DNA, short for deoxyribonucleic acid, and that is passed from parent to offspring. The cell of every organism contains a specific number of chromosomes. For example, humans have 46 chromosomes, and a carrot only has 18 chromosomes. Chromosomes have two parts: the chromatids that are held together by a centromere. And when the chromosome is duplicated after the S phase of interphase, it looks like an X because there are two sister chromatids. They're the exact same genetic information on either side. So the centromere is the circle in the middle, and the chromatids are either the left or right side of the X. And together they make a chromosome. They're exact copies of each other, and they're split apart during mitosis to form single chromosomes. Sometimes these are called double DNA. So the process of chromosome replication starts with one chromatid, and it's replicated during the S phase of interphase into two sister chromatids held together at the centromere. When cells divide, sister chromatids separate so that each cell has the correct number of chromosomes. If you look at the top, we start off with one unduplicated chromosome, or one chromatid. Then we duplicate it during the S phase of interphase, and through mitosis, we split those sister chromatids apart into two separate cells. So at the end, you're left with two cells that have the exact same genetic information as the parent cell. So basically making a clone. So when we talk about diploid and haploid, it's 46 or 23. Diploid, or diploid sometimes it's referred to, are cells that have the full number of chromosomes. So 46 full chromosomes if you're a human. It's different for different species. It's just a full number of chromosomes. You have 23 homologous pairs of human chromosomes. You get 23 pairs, so 23 2. You have one from mom, one from dad. So that makes 2 of chromosome number 1. And you have 2 of each of the chromosomes all the way through 23. One from mom, one from dad. That makes a diploid cell. They are your somatic cells, or body cells. We talked about the fact that the prefix som, S-O-M, means body. and they are every cell in your body except your sperm and egg. Haploid cells have haploid half of the number of chromosomes. So they have 23 individual chromosomes. They have one of all of the chromosomes, either from mom or dad. They are genetically different, okay, switches. And they are your gametes or your sex cells, your sperm and your egg. Human diploid chromosomes have 46 chromosomes, 2 copies of 23 different chromosomes, and the picture of all the chromosomes with their homologous pair is called a karyotype. We saw this when we watched the crash course video on meiosis, and we spoke about the karyotype. So you have 2 copies of each chromosome, starting with pair number 1 and ending with the pair number 23, which are your X and Y chromosomes, which determine your sex. Female has 2 X's, and a male has an X and a Y. The cell cycle for somatic cells, that's your body cells, has 2 main parts, and they are interphase and mitosis. Interphase is everything in yellow, and mitosis is everything in green. You can consider cytokinesis, the division of the cytoplasm, as part of mitosis. It happens at the same time as telophase. So interphase is the longer part of the cell cycle. About 90% of the time cells are in interphase, and it has 3 phases in it: G1, gap, S, synthesis, and G2, growth. And cyclins are proteins that control the cell progression through the cell cycle. So when a cell starts to get too large, it's growing, it sends a signal to the cell through these cyclin proteins that says, okay, we're getting a little too big, we need to divide. So they start the process of synthesis and making copies of the organelle, and then going through the entire mitosis process, and it is controlled by proteins that spur it on through all the different stages. During interphase, DNA is relaxed and spread out thinly, and that is called chromatin. Chromatin is invisible, so it ends in an IN. You can think of invisible. When you look through a regular light microscope, you cannot see it. It's thin, stringy DNA. So the 3 phases of interphase: G1, the cell's living and doing its normal processes. S phase, synthesis, is where the DNA replicates or duplicates. They mean the same thing. G2 is growth. The cell is expanding, replicating organelles, and getting ready to divide. And like I said before, there's no turning back after the S phase. The cells will go through division. After interphase, we enter mitosis and cytokinesis, which is the cell division. And mitosis is nuclear division, which is the division of the nucleus, and cytokinesis is cytoplasmic division, which is division of the cytoplasm or the fluid that's inside the cell through the creation of the cell membrane. We call both together mitosis. So why do cells divide? You should be able to answer that. There were 3 reasons. If you cannot remember them, it was also on your review sheet. We talked about it in class, but go ahead and go back and try to figure that out. Growth, repair, replacement, reproduction. Okay, this is what I hope you were able to come up with. For mitosis, it's growth, repair, replacement, and for meiosis, reproduction. In mitosis, diploid somatic cells divide to become, are they going to be diploid or haploid? They're going to be diploid. And the daughter cells, are they going to be somatic cells or gametes? Somatic. So mitosis makes exact copies of itself. So you start with a diploid somatic cell and you end with 2 diploid somatic cells. Mitosis has 4 main phases plus cytokinesis, and they are prophase, metaphase, anaphase, and telophase with cytokinesis. We call it PMAT. And the hand signals, again, I showed you in class: prophase, metaphase, anaphase, telophase. So we have this table. You already have it printed out. If you were absent that day and you do not have it, then go ahead and make a copy of it for yourself. Prophase is when the chromatin thickens into chromosomes, the nuclear envelope disintegrates, and centrioles migrate to opposite poles, and they start making the spindle fibers. So the centrioles are the little two little pairs of yellow organisms there, and the spindle fibers are the strings that are coming from them. The chromosomes here are in the middle, and they look like X's, and you can see the nuclear envelope, which is the membrane that surrounds the nucleus, is starting to kind of like dissolve or break down. And that's prophase, so we represent it like this. Remember, your fingers are the chromosomes, and since they are duplicated and paired up, here, duplicated, you've got two. In metaphase, they line up in the middle. Middle. Remember, spindle fibers are made of microtubules, and they attach to the centromere of the chromosomes. They line up along the metaphase plate, middle. In anaphase, the chromosomes are going to split apart. The sister chromatids are going to be pulled to opposite sides. The spindle fibers shorten, sister chromatids go to opposite sides. So they were an X, and now they're going in opposite directions. And in telophase and cytokinesis, the nucleus reforms, and the chromosomes relax back into chromatin, back to G1 phase, and the cytoplasm splits to separate the daughter cells. So in animal cells, when the cytoplasm is splitting and new cell membranes are forming, it's kind of pinching off, okay? And that we call a cleavage furrow. That pinching in is cleavage. And in a plant cell, we have a cell wall, so it doesn't pinch in, it can't, it's rigid. So you form a cell plate in between, which is where the new cell wall will be. So here are a couple pictures of an actual slide of cells that are in the process of dividing. And so try to practice seeing if you can tell what phase they're in. Again, here is just an overview of the entire cycle. And remember the acronym I taught you. Insane purple monkeys are terrible comedians. Ipmatic. You need to know the phases. You need to know them in order. Some things you've just got to memorize. You need to know what's going on. So we've got insane purple monkeys are terrible comedians to help you remember the order. Ipmatic. Also interphase, prophase, metaphase, anaphase, telophase, cytokinesis. That's like your little cheer. So hopefully you have enough there to help you remember the phases of mitosis and do well on your test.